In this video, we're going to review how to complete Punnett squares that look at different inheritance patterns. So we're going to start with a simple monohybrid example. So let's cross two cats. Let's say that black fur, which is represented by capital B, is dominant to brown fur, which is represented by lowercase b. And let's assume that we're crossing two parents. One of the parents is heterozygous for their fur color, so they have the genotype capital B, small b. And we're going to cross them with another cat that has brown fur, so its genotype is small b, small b. So when we make our Punnett square, we're going to put a capital B on one column, a lowercase b on the other column, and this is of course because this parent has those two alleles in its genotype. And then we're going to put small b on the two rows on the other sides of the box. So then you're going to fill out your Punnett square. So you should have capital B, small b, small b, capital B, small b, and small b. That gives us these potential offspring. So these are what their children might look like. And what that shows us is that 50% of their offspring will be black cats, and 50% will be brown. Now let's look at an example that goes over incomplete dominance. So remember, when something has incomplete dominance, there is not a trait that's dominant or recessive. You have two traits, and when they are expressed together, they give you an intermediate phenotype between one trait and the other. So an example might be like snapdragons, which are a type of flower. So if we have snapdragons, the letter R is going to represent red flowers, W is going to represent white flowers, and when we have different genotypes, we can look at that. So if you have capital R, that's going to give you red flowers in this species. If you have capital R, capital W, that's going to give you the intermediate phenotype pink flowers, and WW is going to give you white flowers. And so you can see that the red allele does not fully hide the white allele, and the white allele does not fully hide the red allele. So that's why this is incomplete dominance. So in our example, we're going to cross a heterozygous pink flower with another heterozygous pink flower. They both have the genotype RW. So we're going to put the genotypes on the top and the side of the box, and then you can fill out your Punnett square. And when you do that, you'll notice that 25% of the flowers are going to be red because they have the genotype capital R, capital R. You'll notice that 50% of the flowers are going to be pink because they have the genotype RW, and then 25% of their offspring are going to be white because they have the genotype WW. In this example, we're going to look at an X-linked or sex-linked trait, and specifically a recessive trait called hemophilia. So if you recall, hemophilia is a disease where people have limited ability for their blood to clot, and so it can cause excessive bleeding and easy bruising and things like that. So hemophilia is carried on the X chromosome. So in our example, we will have the X that carries a normal clotting factor, the X capital H, and the hemophilia X, or the affected X, will be X lowercase h. And remember, females have two X chromosomes, males have one X chromosome, as you can see here. And so males are more likely to be affected by X-linked traits because they don't have two X chromosomes where one trait can hide behind a dominant trait. The Y chromosome does not hide traits that are found on the X chromosome. Let's look at a couple of the genotypes that are possible for hemophilia. So if you have a female who has X capital H, this is a normal clotting female, so she clots normally and she is not a carrier for hemophilia. If you have X capital H, X small h, this person is a carrier, but they still clot normally because their X capital H allele allows them to have normal blood clotting. If you have X lowercase h, this is a female who has hemophilia. She does not have a normal clotting gene, and she has 2 copies of the hemophilia gene. If you have X capital HY, this is a male with normal clotting ability. He does not have hemophilia. And if you have X lowercase hy, this is a male who has hemophilia. So next, let's do a Punnett square for the cross that is listed above. So we'll put X capital H, X small h. So we're going to cross a female who's a carrier for hemophilia with a male, X capital H, who does not have hemophilia. And we're going to find out what the chances are that their children might have hemophilia. So let's fill out our Punnett square. And as you can see, they have a 50% chance of having a male child, 50% chance of having a female child. And if you look, you'll notice that one of their children is going to, or one of their potential children is going to be a female who is not a carrier for hemophilia. She has 2 normal clotting factors, so she does not have hemophilia. They also have the chance of having a daughter who's a carrier for hemophilia because she has 1 capital H and 1 small h, but she also does not have the disease. They also have a chance of having a son or a male child who has a normal clotting factor. So this is a male who is not a hemophiliac. But because the mother is a carrier, they do have the chance of having 1 child potentially who is a male who has hemophilia because he has that hemophilia allele. Before we move on to doing a Punnett square that shows multiple alleles and codominance, let's review some information about blood groups or blood types. So if you recall, blood types can have different proteins on the surface of blood cells. And so in humans, the most common blood groups are type A blood, type B blood, type AB blood, or type O blood. And those are coded for by a genotype. So if somebody has type A blood, notice that they have little proteins on the surface of their blood cells. That's what makes it type A blood. If someone has type A blood, they can have a couple different genotypes. Type A blood can have an AA genotype or an AO genotype. And the reason that they could be AO is because O does not have surface proteins the way that A does, and so it allows a person with type A blood to express just the A antigen, and then there's no additional proteins that come from that O allele. You can also have type B blood, and again you can see there's different proteins on the surface of the blood cell that make it type B that are different from type A. The genotypes that are possible for a person with type B blood include BB or BO. If you have type A blood or AB blood, there's only 1 possible genotype, and that's because in AB blood we have both type A proteins and type B proteins on the surface of the blood cell. In order for that to happen, a person has an AB genotype, and that way their body will develop A and B proteins on the surface of their blood cells. And finally, if someone has type O blood, notice there's a lack of proteins on the surface of the blood cell, and that makes it type O. And the genotype for type O blood is simply OO. They cannot have an A or B allele and still have type O blood. So knowing all of that information, we can review now how to do a Punnett square involving blood type. So in this example, let's cross 2 people. We have a person who has type A blood, but they're heterozygous for their type A blood, so they have the genotype AO, with a person who has type AB blood, so they have an A allele and a B allele. So when we make our Punnett square, we'll put 1 parent at the top, we'll put 1 parent on the side, and then we'll figure out what their potential offspring look like. So they're going to have one child who has type A blood, who's homozygous for type A. They also have the potential of having a child who is heterozygous for type A, so their genotype's AO. They also have the chance of having a child who has AB blood, because they have both an A allele and an O allele, or B allele, sorry. And they also have the chance of having a child who has type B blood, because they can inherit that B allele and an O allele. So in this cross, there's actually 4 different genotypes possible and 3 different blood types possible. So you'll notice that half of their children, or 50%, have type A blood, and then a quarter have type AB blood, and a quarter of their children have type B blood. Finally, let's look at an example of a dihybrid cross. So here we have a cross between some plants. So we're going to say that capital T means that it's a tall plant, lowercase t means that it's a short plant, capital A is an orange flower, and lowercase a is a white flower. So we have a couple parents here. One of them is heterozygous for height and heterozygous for color. The other is homozygous tall and heterozygous for its orange color. So the first thing we need to do is figure out what the gametes are. So remember, you're always going to combine one allele for height with one allele for color. So for our tall and orange plant that is heterozygous for both traits, we can have capital T, capital A. We can have capital T, small a, which would be here. We can have small t, capital A, and small t, small a. So those are the gametes for the first parent. Now let's look at the gametes for the second parent. The second parent has 2 copies of the tall allele, which means they only produce 2 different types of gametes. So you can have a capital T with a capital A, or you can have a capital T with a small a. Because they have 2 copies of the same allele for tall, you could have 4 gametes, but they are redundant. So we only need to write it twice, right? So we just have capital A and capital T, capital A, and capital T, small a. Next, we'll fill out our Punnett square. And now that that's done, you can see that we have a few different traits that we have inherited, or different combinations. So you could have offspring that are homozygous for both dominant traits. Sometimes you'll have something that's homozygous for the dominant trait, but heterozygous for color. Notice though that they all have a capital T. Because this parent does not have a lowercase t, all of these individuals will be tall, but some of them will be orange, which is the capital A. So we'd have an orange one here, orange here, orange here, but some will be white. So there's a couple plants that will give us white flowers.